Today's screencast is about Chapter 13.1 and 13.2, the replication and structure of DNA. There were multiple people searching for the genetic material and trying to figure out whether it was protein or whether it was RNA. We knew about all these different types of materials, but we didn't know which one was the hereditary material. So Griffith in 1928 found that genetic material could be transferred between cells. Avery found that DNA was a transforming agent, and Hershey and Chase found that DNA is the hereditary material rather than protein and rather than RNA in bacterial cells. Also, at about the same time in the 50s, Rosalind Franklin had photographed DNA with X-ray crystallography, and Watson and Crick were able to get a hold of her X-ray picture and determine that the DNA model was a double helix. As you can see from the model, the structure is basically a long, twisty, rope laddery kind of thing. So the main shape of the DNA molecule is the form of a spiral staircase or even a twisted ladder. It's also known as a double helix. What is a nucleotide? Watson and Crick had found that the actual materials that the DNA is made out of are called nucleotides. They called them that. They're the subunits that make up DNA. There are three main parts: a phosphate group, five carbon molecule, sugar molecule, and a nitrogen-containing base. And there are different numbers of hydrogen bonds between each of the kinds of bases. So what is deoxyribose? It's the 5-carbon sugar that's found in the DNA nucleotides. Deoxyribose is what DNA is named for, deoxyribonucleic acid. What are the DNA base pairing rules? In DNA, adenine always goes with thymine, and guanine always goes with cytosine. So how do the bases line up? First of all, they are complementary. So you have certain bases on one side of the strand, and then you add the other bases. The sequence of the one strand determines the sequence on the other strand. What is DNA replication? It's the process of making a copy of DNA for passing on the genetic code. This is the part of inheritance that everybody was looking for. So during interphase, you have a single strand of DNA and you have two strands on each side, and then what you do is zip it open and add the complementary bases. Then you end up with two exact copies of the same molecule. So what is helicase? Well, it's kind of like a pair of scissors. It's an enzyme that opens up the double helix by breaking through the hydrogen bonds that connect the bases together. So it kind of snips it open. A replication fork is the Y shape that's created as the double helix is opened up. What is DNA polymerase? Well, it's the molecule, the enzyme, that move along the strand and add the complementary nucleotides together. So here's the summary. DNA that's to be replicated, the strands separate and open up using helicase, and then each complementary side has bases added to it, and that's using polymerase, and then there are two copies of the original DNA.